Hi everyone, good afternoon, good evening. Welcome to this OSME lecture on neurodevelopment and neuronal function. I'm Harry, and please do shout any questions into the chat. The outcomes for this lecture today are to understand the key principles of embryonic development and neurodevelopment in humans. And I'm going to split that into two parts, really. An early part looking at kind of the course processes involved in neurodevelopment, and then a more detailed look into some of the molecular and cellular processes that are going on. We will then move on to that second part of the lecture about the neuronal function part of your syllabus. So as I kind of said, this is the outline, and most of our time this evening will be looking at neurodevelopment, because I think it's the most complex area and the part that people probably have the hardest time understanding. So I apologise if the neural function part at the end is a little bit rushed, but I hope that I've provided some decent notes that you can use for your own learning. So this is the neurodevelopment part of your syllabus. If you're in Oxford, if you're not in Oxford, I am sorry for leaving you out. Brilliant. So let's start with kind of a broad overview with CNS development. And when thinking about any developmental process, I think it is really important to think about what is going on at the level of individual cells and tissues in that developing system. So whilst I'm giving you the lecture today, I would recommend that you just think in the back of your mind what various developmental processes are going on. What are the cells doing? What are the tissues doing? And maybe even come back to this slide at the end and try and allocate some of the processes that we've spoken about to some of these cell level processes. I always found this idea quite tricky during my first and second year. So if you have some questions about it at the end, I'd be very happy to take those. But it's useful, I think, for thinking about your essays and things, because often you'll get a curveball essay question that asks, for example, you might get a question that asks how mitosis and migration contribute to neurodevelopment. Now, that is simply asking about the process of neurogenesis, as we will see, but it phrases it in a different way and gets you to think about it a bit differently. And so hopefully I will help you to think about those processes and to join them up together in this lecture. But without further ado, let's get started, because there's a lot to go through. And really the story with neurodevelopment begins after gastrulation. So obviously you will remember from your first year that gastrulation is this process whereby a bilaminar germ disc becomes trilaminar, and we establish the three primordial germ layers of the developing embryo. Those are the ectoderm, the endoderm, and the mesoderm. Not in that order, though. The mesoderm's in the middle. And that's kind of what we see in this small diagram up here around day 16. In terms of thinking about a neurodevelopment essay, please don't describe gastrulation in great detail. I will cry for your hands if you're writing that much. But just be aware that gastrulation has happened, have that definition in mind, and then you can really start your essay assuming that gastrulation has happened. So what does happen after gastrulation? And for neurodevelopment, we are really interested in the ectoderm. So the entire nervous system is ectoderm derived. So the ectoderm is this top layer of the trilaminar disc in blue, and we get two processes going on. The first being neural induction, which is where the mid aspect of this ectoderm tissue thickens and establishes what we call the neural plate. That neural plate receives signals from the notochord, which is this structure underlying it here within the mesoderm, to fold up, to proliferate, for cells to change their shape and to roll up into a tube. We can see that continuing down here at around day 20. And then by embryonic day 22, that tube is closing up dorsally, and you are forming a complete neural tube. And that is the process of neurulation. And that neural tube is going to form the basis of our nervous system. So here's the neural tube a little bit bigger. Once it's completely closed, the neural tube kind of becomes completely separated from this overlying ectoderm and sits within the mesoderm. It is flanked on either side by your somites. It is flanked ventrally by the notochord. Those three, the somites and the notochord, they're mesodermal structures. And as you will recall from your first year embryology, they are important in forming the vertebra and the bones of the spinal column. We won't really talk about them today. So thinking about that neural tube, as we spoke about in the previous slide, the neural tube closes from that from the neural plate, and the first part of it to close is the middle part, and then the closure of the tube progresses in a rostral direction and in a caudal direction. And we say that the formation of this neural tube is really critical in the establishment of that rostral caudal axis. Now, what does rostral caudal mean? Rostral means nose, caudal means tail, kind of relating to your coccyx, I guess. It's basically an overly complex way of saying the body axis that runs from head to toe. Obviously, a body axis in development is it's talking about an asymmetry within the embryo. So the important ones we talk about are rostral caudal, head to tail. You kind of have left, right, or medial lateral. And then you have your dorsal ventral, which is kind of front to back or anterior posterior. Brilliant. So you get this midline closure, progresses towards each end, towards the rostral end and towards the caudal end. And eventually you are left with these kind of openings at either end called your neuropores. So this neural tube that we have formed by neurulation is going to form the basis of our nervous system, as I've said before. The wall of the tube is we call it the neuroectoderm tissue, and those are the cells that are going to become all the cells, the neurons, the supporting cells of our brain and nervous system. And then the lumen of the tube is going to become the ventricular system of the brain and the spinal canal. Obviously, you know, we love to talk about developmental disorders and problems that arise during development, and a really important group of those thinking about neurodevelopment are your spina bifida diseases, which are developmental problems where you do not get the complete closure of the neural tube. So obviously there are varying degrees of severity of these. I hope these are words that you recognise. So you get spina bifida occulta, which is really a more minor defect, whereby the problem really is only in the complete, a complete vertebra fails to form, progressing all the way up to myelomeningocele, which is where you get protrusion of neural tissue completely outside the body wall. And obviously on that scale of severity will become kind of more debilitating defects. That's kind of thinking about the caudal end and the spinal cord. If this failure of closure happens in the cranial area, then you're really going to get this more severe disorder called anencephaly, which is where you get incomplete formation of parts of the skull and parts of the brain, and there are going to be quite serious neurodevelopmental consequences to that. A really important piece of work was this in red at the bottom in 1991, showing that folate supplementation was sufficient to reduce these types of neural tube defects, indicating a role for folate in, that process of neurulation. Maybe we can have a think about what that role is a bit later on. And now folic acid supplementation of numerous foods is widespread, including in the UK. So we've got a neural tube, and we now need to pattern that neural tube to create the kind of shapes and the structures of the mature nervous system. So in embryology, we call that process patterning. And focusing initially on the cranial head end of the neural tube, we see these kind of expansions of the neural tube called your primary brain vesicles. There are 3 of these, and they correlate roughly to the major anatomical areas of the brain. Those are the forebrain, the midbrain, and the hindbrain. These primary vesicles, as they grow and as they expand, also become divided, and that's how these 5 secondary brain vesicles emerge. These are the telencephalon and the diencephalon, which constitute the forebrain, the mesencephalon, which will become the midbrain, and the metencephalon and the myelencephalon, which are hindbrain structures. Really, you know, there is quite a lot of words really to learn here, and it is probably going to be useful for you to know, have a rough idea of what structures are derived from these vesicles. So I have provided a list here that you can use in your revision, and the anatomical divisions as well. So we've got our brain vesicles and we're also going to have a process of folding of the neural tube in order to bring these vesicles into the anatomical, gross morphological arrangement that we see in the adult brain. The first of these flexures or folds to emerge is the mesencephalic flexure, here in orange, which is between the forebrain and the midbrain. We then get the pontine, the cervical flexure, sorry, getting ahead of myself, here in green, which occurs between the top of the spinal cord and the rhombencephalon / hindbrain. And these are both folds in the ventral direction, so folding anteriorly towards the front of the embryo. We also get this dorsal or posterior fold called the pontine flexure, and that is within the hindbrain, separating the metencephalon and the myelencephalon. Now, what are the consequences of these folds? Well, this is quite a complex diagram. I wouldn't worry about knowing every single detail of this, but I hope it will illustrate some of what I'm talking about. So here are our flexures in yellow, here are our primary vesicles in green, and here are our secondary vesicles in pink. So really thinking about kind of the shapes and the morphology of the mature brain, I'm going to start at the forebrain here, and we see the telencephalon expanding and folding like a pair of great big bunny ears over the diencephalon, which sits immediately beneath it. So this is these are both forebrain structures, and we get the telencephalon, which has these 2 big bunny ears, if you like, and they will eventually form the 2 hemispheres of the cortex. They fold down and sit atop the diencephalon, which is kind of gives rise to important subcortical structures like the thalamus and the hypothalamus. So you can picture that thinking about your anatomy. You then get the midbrain structures just beneath that, of which the superior and inferior colliculi are important mesencephalon-derived structures. Moving into the hindbrain area, and we see this pontine flexure, which is the posterior fold that we were talking about, and that is really important for bringing the metencephalon, which will give rise to the cerebellum and the pons, into its definitive position at the back of the brain, posteriorly, and in effect sat almost on top of the myelencephalon, which forms the medulla oblongata and the brainstem. So again, a lot of words in this section. Do revisit it, and hopefully some of those words will fit into place and make sense. So if some of you are logged into Socrative, I hope you are, you can attempt this question on your phone. If not, I will leave it up on the slide here. Let me have a look. We have 6 students in Socrative. So do have a go at answering this one. From which secondary brain vesicles do the optic cups derive? Now, this is really harsh of me because I actually didn't explicitly say, but if you were looking at my diagrams, and you might be able to remember. All right, so I've got 8 out of 10. I'm going to show you the answers there. Brilliant. 60% of you on Socrative got the diencephalon. That was harsh of me. I didn't quite explicitly get that across, but if you look on this slide here, which I hope you can still see, these are those bunny ears that I was talking about of the telencephalon, the diencephalon sitting just underneath that, and this gives rise to those subcortical structures, and you can see the eye cups either side that will become the retina. Brilliant. Next question. This is a hard one with lots of words. Sorry. That second question should now be appearing on Socrative. I would hazard that you would probably be really hard done to get a question this detailed in your exam. You know, my justification is that I made these questions up because I couldn't really find a bank of decent neurodevelopmental ones, which is probably no solace to you, but you know, just think through it logically. Got 6 out of 10. I'm going to show the answers. Amazing, 67% did get that right. Oh no, someone's just changed. Don't worry. Yes, that was a real harsh one. The medulla oblongata is derived from the metencephalon. For those of you, sorry, I didn't realize those of you who are in the slideshow there. So yes, the medulla is the metencephalon, which is the caudalmost of those secondary vesicles. Is from the myelencephalon, sorry. The metencephalon is the next one along, and that's the one that's forming the pons and the cerebellum. Don't worry about those, they were cruel. Brilliant. So that's it for the questions in this part of the lecture. I'm now going to try and move on to a little bit of kind of cellular and molecular detail about neurodevelopment. I think this is by far the most interesting part of the lecture. So if I go off on one about something I find exciting, then I'm really sorry. So the production and the birth, if you like, of new neurons is a really important part of the development of the central nervous system, and that process is neurogenesis. Now, by the time you are an adult or even a child, we say that your brain is mature and that neurons are not proliferating, which is inconvenient at best. Anyway, so during development, you get a huge amount of mitosis to produce many millions of neurons that will eventually form your brain and your spinal cord. This neurogenesis takes place in the ventricular and subventricular zones, which I will show you shortly. They say that your peak neuron count occurs at 18 months old, and that you lose 1,000 neurons a day from that point onwards, which, don't think about it too much, it's distressing. But it's important to emphasize that this loss of neurons is a natural, normal physiological process, and it is intrinsic to kind of the maturation of the brain and the development of mature circuits, to learning, and also to healthy aging. But obviously there are circumstances where this can become pathological. Having said all this, and whilst the bulk of neurons in the mature brain do not proliferate, there are small niches in the adult brain capable of producing new neurons. And these are the subgranular zone, which sits within the dentate gyrus of the hippocampus. And that's quite a mysterious little area, actually, and there's a lot of debate about what it does. It's obviously in the hippocampus, so it's questioned as to whether it has some role in the formation of memories, or even kind of the way that memories are encoded and consolidated after you've learnt them. But a lot of debate about that. The subventricular zone of the lateral ventricles is the more known about area, and the neurons born here undergo a rostral migration, through this rostral migratory stream, and they provide the neurons that are replenishing the sensory neurons in the olfactory bulb at the top of the nose. These niches are of academic interest, including in Oxford, because, you know, the dream would be that we could target these areas to provide new neurons in damaged brains, say after a stroke. I would hazard that that is a long way off. And here is a very beautiful image of, I think, a rat brain. And you can see the lateral ventricles here, the black areas, and you can see a huge amount of proliferation marked in pink here in the ventricular and the subventricular zone lining that ventricle. You can see those neurons migrating rostrally into the olfactory bulb, which in a rat is absolutely massive. The olfactory bulb is not that big in humans. You can also see a little bit of proliferation happening in the dentate gyrus, and that's the area that is a little bit more mysterious. So what do we mean by the ventricular and the subventricular zone? The ventricular zone is this bottom part of the neuroepithelium lining the ventricle walls. So I want you to picture this as a slice, this entire image as a slice through our neural tube, just like this. We have the pia, which is the outer coating of that tube and eventually of the brain. And we have the ventricular zone, or the ventricular surface down here, which is lining the lumen and the ventricles. You will sometimes hear these referred to as kind of the apical and the basal surfaces. And it's a bit counterintuitive that proliferation takes place in the apical area, whereas in most epithelia, skin, for example, we associate proliferation with the basal area. But I need you to think about this as a flat epithelium that during neurulation has rolled up to form a tube. So the basal surface is now around the outside, and the apical surface is the middle, and that is where proliferation is happening. So what's the mechanism for neurogenesis? Well, we get these radial glial cells, which are the precursors to glutamatergic excitatory neurons in the central nervous system. They undergo mitosis in the ventricular zone, and they also do this really interesting thing, this kind of nuclear translocation, which is where their nuclei translocate from the ventricular zone up to the pial surface for maturation. We don't really know. We do know that they establish these glial fibers that are later useful for the migration of neurons, but then the nuclei translocate back and they undergo mitosis at the ventricular zone before migrating into this mantle zone, which will kind of when the neurons will happily sit and mature, and where our nervous system will develop. So how does neurogenesis occur? We have a direct route whereby one radial glial cell divides, it produces another radial glial cell, and it produces an immature neuron that migrates out into the mantle zone. We also get this indirect route whereby one radial glial cell divides into two what we call intermediate progenitors. These are quite interesting. They sit in the subventricular zone for a little while, just above the ventricular zone, before migrating out as immature neurons into the mantle zone. What's interesting about these processes is that many studies have shown that there is no difference in the cell types that these two processes yield. So we don't know why some neurons go directly from the VZ, and we don't know why some go through this indirect route and hang out in the subventricular zone as intermediate cells for a little while. So we have lots of proliferating cells in the ventricular zones and the subventricular zone lining the ventricles. These newborn neurons then migrate radially into the developing cortex. And now this is quite a complex diagram, and you don't need to know the ins and outs of it, but I think it helps me to demonstrate kind of this idea of radial migration. So you have this image, it's from a mouse or a rat, I think. It's been stained with a radioactive marker that marks actively proliferating cells. And you can see that the ventricular zones here have stained really, really heavily. because there are lots of actively proliferating cells in there. You can then see this grey area overlying, which is where cells that are born in the ventricular zones have migrated out into the developing cortex. Now, what's interesting about this image is that you actually get a bit of a time gradient. So for whatever reason, don't worry about this, but just take my word for it. The anterior part of the developing mouse brain over here is a little bit ahead of the posterior part here. Don't know why. Interesting for this diagram. So when we have taken a slice from here, you can see, in effect, in box C over here, the first wave of neurons migrating out of the ventricular zone, and they're forming this structure called the preplate. Then as you follow this round, you can see that the preplate becomes separated, and this clear area in the middle is a second wave of migrating cells that hasn't been stained because they were born later than when we gave the radioactive marker. But you can see that this wave of neurons interrupts the preplate, splits it in two, and then we get the marginal zone on top. We get the cortical plate here, which is those new cells that are going to form the definitive cortex. And then we get this layer underneath called the subplate, which is super, super interesting, and we will get onto. So here's a friendlier version of that with slide animations. So here we go. So your first wave of neurons migrate from the ventricular zone and they form the preplate. Your second wave of migrating neurons sit in the middle of the preplate and they form the cortical plate. In doing that, they divide the preplate into a marginal zone on top and a subplate underneath. So the cortical plate forms the definitive cortex, and the layers of the cortex, as I'm sure has been drilled into you over and over again, form in an inside first, outside last fashion. So the first layer to form is layer 6, the next layer to form is layer 5, and each wave of migrating neurons establishes kind of the next layer out, if you like. So over on the right hand side, we see, if you like, a fully developed cortex where layer 6 was the first to form. We had inside first and then outside last, layer 1 being the last layer to form. And what we see here is that this subplate, which I heralded as the most exciting thing ever, is gone. Most of the cells in the subplate have apoptosed at this point, and the subplate is a transient structure, and it plays a really important role that I will go on to shortly. Another feature of these waves of migrating neurons that we see during neurodevelopment is that each wave, each layer, carries its own signature gene expression, if you like, and expresses a unique set of transcription factors that define each layer of cells. So that is useful for two reasons. One reason is that those transcription factors are actually cross-inhibitory on one another, meaning that cells in one layer, transcription factors, I should say, sorry, in one layer will antagonize the transcription factors expressed in the next layer, and that means that they can't be co-expressed, and you get these really clearly demarcated boundaries between your layers, which, you know, neuroscientists love and draw no end. So kind of just to try and explain this, and this is quite complex experiment really, but just take my word for what I'm about to say. So SOX5 is one of our genes here. I'm going to tell you that it specifies the subplate. So SOX5 is required for neurons to be part of the subplate, and it inhibits this marker CTIP, which is a transcription factor expressed in layer 5. So if, as in panel B here, you knock out SOX5 in a knockout mouse, you get no subplate forming, and the layer 5 neurons are completely disorganized. And that's why, you know, this idea of like cross inhibition of the transcription factors in the cortical layers is useful, you know, to help convert those waves of migrating neurons, which is a kind of temporal code, if you like, into a place code, which is genetically defined. Finally, these transcription factors are useful in that they determine the neuronal fates within those layers and determine what type of neurons you're going to find there. And the obvious example is that kind of the genes expressed in layer 5 will cause these neurons to terminally differentiate into projection neurons, which form those long projections out of the cortex towards the thalamus and other subcortical structures. So we've said that this migration of neurons is really important for the developing brain. And so therefore, we're obviously going to find a developmental defect where that doesn't happen. So Lissencephaly describes kind of a family of diseases associated with abnormal migration of neurons, and that results in the formation of a smooth brain. Now, I need you to remember at this point that the folding up of the cortical surface to form sulci and gyri isn't like a patterned or like a program, if you like. Rather, it's simply a function of an expanding surface area in a limited amount of space. So as you get ongoing neurogenesis and proliferation, and then migration outwards of those neurons, the cortex is going to expand, increase its surface area, and fold. Now, in Lissencephaly, when where you get these mutations that prevent normal migration, you get a disorganized cortex, a thin cortex, and you do not get this normal folding, as you can see in the CT images over here. This is just an example of kind of a gene mutation that this Lis1 gene here that causes a failure of migration and a real jumbling up of the cortical layers, and also a smooth brain. Reelin again, reelin is really interesting. I'm not going to go into it in huge detail. But if you have a reelin mutation, your entire cortex is flipped. So it forms outside first, inside last, which is just cool. Zoltan Molnar always goes on about it. I'm going to go on about it here. So I promised you that I would talk to you about the subplate, this really important, albeit transient structure at the first layer to form in the cortex, the insidemost layer, and a layer that is eventually lost. And what I'm going to explain to you is that the subplate acts as like a scaffold and is really important for the organization and the development of all the other cortical layers. So the earliest connections between the cortex and the thalamus are actually with the subplate, and growing neurons from the thalamus form their first synapses in the subplate. The subplate is then really critical for remodeling those circuits and those synaptic connections and establishing kind of the definitive circuits in the cortex. And that's not only a process of kind of plugging in the neurons to the right place, but also this process whereby electrical activity within the cortex helps to carve out neural circuits. And then once that job is done, the subplate is a transient structure, and it does not persist into adulthood, other than forming these funny little white matter tracts at the very bottom that nobody really knows what they do. So this is just a slide to reiterate that. What's interesting on here is the fact that if you do a GWAS study, which is looking for genes associated with specific diseases, you find that neurodevelopmental disorders such as autism, ADHD, and schizophrenia are all associated with genes that are expressed in the subplate, which indicates that the subplate is really, really important for the formation of proper, fully functioning neural circuits. And this image in red is very much off syllabus, but it's just to show you really, this really profound ingrowth of axons into the subplate before forming these projections. And this effect is so strong that in that Rila mouse that I spoke about ever so briefly, where all the cortical layers are flipped, the axons will still track all the way up to the top for the subplate and then dive down and form this looping sort of effect. Very cool. So we've talked about the subplate and what other ways are there for neural circuits to be formed and sculpted out. And this is really a very brief slide, and I'm sorry I can't go into this in more detail, but we are time limited, I'm afraid. So what's going on in the brain? We get molecular patterning, which is where you get molecules, chemical gradients across the entire cortex, and they help to, you know, define the fates and the functions of different parts of that cortex. You get some more complex systems contributing to the establishment of neural circuits, and those lectures that you had on the development of the retinal, the optic tracts and things like that will be really, really interesting for you to go and look over after this. Just to flag briefly what I mean, you get two systems going on here. You get this efferent B system, which is where these neurons expressing FB are repelled at the optic chiasm by efferent B, and that causes them not to cross the midline and be projected to the superior colliculus, this is, on the ipsilateral side. And you also get this process whereby you see gradients express of efferent A's in the retina, and that causes their projections to target specific areas of the superior colliculus, which express the kind of complementary efferent gradients. So this is kind of showing that actually the growth of axons and the molecular programs that are guiding those processes are actually really important in the eventual kind of establishment of mature circuits. If you have more questions about this, I'd be very happy to go into it, but we are a bit time limited today. You also get this process whereby the activity, the electrical activity within circuits can actually help shape them. And that's quite complex again. Think of kind of synaptic plasticity sort of processes, and the idea that you're going to form lots and lots of connections during development, and those which are used most are going to be maintained into adulthood. So we've talked a lot about excitatory cells and glutamatergic neurons. We need an honourable mention at this stage to inhibitory interneurons, which are these GABA expressing cells. And these guys emerge in the ganglionic eminences down here at the bottom, and they undergo a tangential migration into the cortex. Now that's very different to the radial migration of the glutamatergic neurons. And what does those terms radial and tangential mean? Well, it basically means that they're migrating at right angles, and they're going to meet in their definitive positions in the cortex, and that kind of meeting and the interactions that they form are going to be really important to carving out neural circuits. The ganglionic eminences also contribute interneurons to other subcortical structures. All right, so we have a couple more questions. Here we go. Pop over onto Socrative. I'll also leave the slides up here so you can see them. I hope I haven't lost too many of you. Got one answer. All right, we've got 6. I'm going to show you the results here. So yeah, absolutely smashed that one. 86% getting the correct answer that the ventricular surface is the site for neurogenesis in the developing brain. One more question. Should be nice and quick. Remember, the subplate is that structure at the, the first cortical layer to form, and we call it the transient or dynamic scaffold. Amazing. So a good proportion of you identifying that it's the subcortical white matter tracts there, 78%, that's brilliant. This, most of the cells apoptose and they form this funny remnant structure that we don't know much about. This is a very wordy one, I am sorry. Brilliant. Seven answers again. Awesome. Oh, all right. All getting the answer as A there. Do be prepared for a horrible wordy question in your exam that makes you question everything you know, just like this one. But vast majority of you absolutely right. Ganglionic eminences are the source for those GABAergic inhibitory interneurons, and they migrate tangentially, which is the perpendicular to the radially migrating glutamatergic neurons. Amazing. Right, let's power on and have a brief delve into the spinal cord, and then we'll get onto the neural function aspect of this lecture. Got 15 minutes to go. I hope I can make it. I might be 20 minutes. I'm really sorry. All right, so this is actually a neat example of those kind of molecular gradients patterning the CNS, which I spoke about earlier. Obviously, they're a bit easier to understand in the simpler context that is spinal cord. So in the spinal cord, we get two molecular gradients, which are really important in patterning the cord and setting up different structures. So we get the BMP and Wnt, which are secreted molecules from the overlying ectoderm. And then we get sonic hedgehog here at the bottom, which is secreted by the notochord. These molecules diffuse across the neural tube, and they induce kind of different fates, different neuronal types on their way, if you like, and the exact combination of those signals that cells receive will determine what the cells become. So in the spinal cord, we can think of this really easily as, well, what forms in the dorsal part of the neural tube? Well, we know that we get sensory input via the dorsal root ganglia from peripheral nerves. These enter via the dorsal horns. So the dorsal region here that's receiving lots of BMP and Wnt signals is going to adopt a sensory fate and form the neurons that receive those sensory inputs. By contrast, the ventral area that receives a lot of this sonic hedgehog signal, I should point out that sonic hedgehog is also a really important signal from the notochord in the initial folding up of the neural tube. So it's a powerful boy, and that is in the ventral area here. Lots of sonic hedgehog signal induces the fate of motor neurons, and those are the neurons that grow out via the ventral horn to their peripheral targets and form lower motor neurons. A very brief mention to kind of the brainstem and the hindbrain, and really I want you to think of the arrangement of sensory and motor nuclei as exactly the same, but I need you to account for the fact that in this part of the brain, the fourth ventricle becomes really dilated and expanded and pushes the brain tissue laterally, if you like. So it's a tube and it opens out. And what that means is that the motor nuclei, in red that were ventral, remain ventral and in the midline. And those sensory nuclei here in blue are no longer dorsal as such, but actually more lateral. And that is a useful thing to think about when you come to learn this horrible slide, which is all about, your cranial nerve nuclei. And it can be quite useful to think about kind of the rostral caudal position as an anatomical position, like what cranial nerve are we talking about? What's the nucleus? And the medial lateral position is actually relating to the function of that, that cranial nerve. Is it a motor nerve? Is it a sensory nerve? And kind of from that system, you can begin to think about where these nuclei sit. Let's get rid of that. It's horrible. I'm sorry that you have to learn all that. The neural crest. This is lovely. We love the neural crest. So as you'll remember from your first year, the neural crest are these neuroectoderm cells at the dorsal-most neural tube up at the top here. They receive very little sonic hedgehog signaling from the notochord here, and that causes them to delaminate, undergo a epithelial to mesenchymal transition, which just means that they unstick and become migratory. And then they migrate along these really long and well-characterized migration pathways and contribute to a huge number of peripheral tissues. Now, many of those are more related to your first year embryology learning, but they do contribute really significantly to the peripheral nervous system. And you can see on the right-hand side here a list of, all the ganglia, these peripheral ganglia. Ganglia are collections of cell bodies, neuronal cell bodies outside of the CNS, which are formed by the neural crest, and which kind of then axons and peripheral nerves grow out of those and plug into their eventual positions. And in terms of those, the neural crest is forming your sensory nerves via the dorsal root ganglia. It's forming many of your autonomic sympathetic and parasympathetic nerves and your enteric nerves in your gut and your cranial nerves. So really important contribution from the neural crest to the peripheral nervous system. I really love the neural crest. I'm sorry, I'm a nerd. I'm actually not going to open this question in Socrative because I really want to power through. I will give you a moment to think about it though. Which of the following is not derived from the neural crest? Enteric nerves, cranial nerves, optic nerves, sensory nerves, sympathetic nerves, or the craniofacial skeleton? All right, so the answer here is your optic nerves. As we mentioned many questions back, the optic nerves are formed from the diencephalon. So the diencephalon forms the optic cup, which forms the retina. The optic nerves are outgrowths of the retina, so it's not derived from the neural crest. The craniofacial skeleton is one of those cool non-nervous system things that the neural crest is contributing to. Amazing. So that is the neurodevelopment part of this lecture done. If you have any questions on that, I'm very happy to stick around at the end of this lecture and answer those, because I do realise that there's a lot of content and it can be quite a complex area. I'm now going to power on to the second half of the second part of this lecture, which is on the neuronal function part. I apologise that there's not going to be a huge amount of time dedicated to this, but I hope that the notes that I've provided are useful for you during your revision. And also, I won't be doing any Socrative questions in this part of the presentation, but I have provided some multiple choice questions in your feedback form that you can have a go at, and will hopefully rejig your memory and help your revision. I think this is an area where, it's a massive syllabus area. I can't promise to cover it all. I think we've spoken a bit about this neuronal repair and regeneration over here. I'm going to try and cover the bulk of this red part. I'd recommend a really good set of flashcards or something to help you power on through this, because there's a lot. So let's start by thinking about neurons and what are our neuronal cell types within the nervous system. Well, we've spoken about these, we've met them. There are excitatory neurons, which are mostly glutamatergic. That means they use the neurotransmitter glutamate. And the vast majority of those cells are what we describe as pyramidal cells, which is kind of just describing their shape. So they have this pyramid-shaped cell body. They form these massive dendritic trees that receive lots and lots of input, and they have little dendritic spines, which are kind of the postsynaptic component of the synapse. And I'm sure that you will meet and think about when you do your LTP and LTD revision. They also have myelinated axons, which can be quite long in some places, particularly if they are kind of projection neurons out of the cortex, and the myelin sheath is important for that fast saltatory conduction. By contrast, inhibitory interneurons are really more varied. The vast majority are still those GABAergic interneurons, the ones that we spoke about forming from the ganglionic eminences, but an honourable mention to a small group of interneurons that use glycine as a neurotransmitter. These are really varied in their shapes and their morphologies, depending on kind of where they sit in circuits and what they are contributing to those circuits. And the names are quite descriptive. So you get basket cells, which sit around pyramidal cell somas like a basket. Get chandelier cells, which I assume look like chandeliers. And, there's no need to know those in great depth, but they are very varied. You then get your non-neuronal cell types. These are your astrocytes, which are really, the do-it-all cells, non-neuronal cells. in the brain. So these guys are important parts of the blood-brain barrier. They are important for regulating the extracellular matrix and the extracellular environment in the brain by regulating cerebrospinal fluid composition. In particular, they absorb excess, they take up and break down excess neurotransmitter, and they help regulate potassium ions at the nodes of Ranvier. And they also interact with T cells to help during inflammation and repair. In terms of inflammation and repair, microglia are the real stars here, widely described as kind of the macrophages of the central nervous system. These respond to injury, they clear away debris and damage, and these are able to proliferate in response to injury, and they are the only cell type here, and they are distinct from neurons as well in this regard, in that these guys are proliferating and forming scar tissue when the CNS is damaged. Finally, you get ependymal cells, which line the brain ventricles. The Oxford Handbook says that they have no known physiological role, which I imagine is a bit harsh on them. I'm sure they're doing something. There's some query about whether they help regulate those neurogenic niches that I spoke about earlier, but not a huge amount to learn about those. And then you have your oligodendrocytes, which are your Schwann cells of the central nervous system. So in the central nervous system, your myelin sheaths surrounding your pyramidal cell axons are formed by oligodendrocytes, not Schwann cells. So let's have a whiz through some different neurotransmitters. And I hope that these slides will be useful to you. I've included some kind of pharmacology and some drugs, which I actually won't talk about today, but they're in the notes. And I hope it helps you to kind of make a bit of a note between this lecture here and other lectures that you have on maybe, you know, psychiatric disorders or mood disorders or the basal ganglia and dopamine and things like that, just to help join the dots a bit. So glutamate is synthesized from an amino acid, glutamine. Glutaminases are the enzymes doing that. It's transported into presynaptic vesicles by the vesicular glutamate transporter. Your postsynaptic receptors are AMPA and NDMA, which you will learn about lots in LTP, etc. AMPA is the sodium channel that is kind of the primary receptor mediating excitatory postsynaptic potentials, so kind of evoking action potentials, whereas NMDA is that really interesting calcium channel that actually has this kind of voltage gating function that exists because of a magnesium ion that sits in the pore of the channel. When the membrane is depolarized, that magnesium ion in the pore is displaced and calcium ions can flow. And because of the need for the ligand, glutamate, and this voltage gating, we call this channel the coincidence detector, and it's really important in LTP. We also get metabotropic receptors, which are a family of numerous different types, and they're all G protein coupled. Reuptake is by the glutamate transporter, and here are that's your pharmacology. In terms of GABA, also synthesized from the amino acid glutamine, with this extra enzyme glutamic acid decarboxylase involved. The vesicular glutamic acid transporter is responsible for loading it up into your presynaptic vesicles, and you get two types of receptor here. The GABA-A receptors, which are ionotropic chloride channels. So when these channels open, you get an influx of chloride into the cell, and that hyperpolarizes the cell membrane and therefore creates an inhibitory postsynaptic potential. And then you get GABA-B, which is your metabotropic receptor, G protein coupled, and a large family with many different G proteins involved. Benzodiazepines and barbiturates are the really interesting pharmacological agents acting on GABA. Benzodiazepines are much more widely used nowadays than their kind of predecessors, the barbiturates, but by increasing GABAergic transmission, these drugs are kind of anticonvulsants and anxiolytics as well. And they can also be used as kind of painkillers and sedatives in other contexts. Uptake is by GAP1. Monoamines, this is a family of neurotransmitters named because they all have this shared monoamine structure. Again, synthesized from amino acids. They have this common vesicular transporter, the vesicular monoamine transporter, and then they have unique receptors in the postsynaptic membrane. So these are the dopamine receptors. D1 is excitatory, D2 is inhibitory. Just be aware, if you're answering questions about the basal ganglia and they are asking you kind of, I don't know, which part of the direct or indirect pathway is inhibitory, which is excitatory, they will love in multiple choice to ask that actually as what's the receptor, and it's just asking you to make a last little jump and remember that D1 receptors are GS coupled and excitatory, and D2 receptors are GI coupled and inhibitory. Then we get our catecholamine receptors for noradrenaline. These are the same culprits that you met no end during your first year pharmacology, alpha and beta receptors, all metabotropic G protein coupled receptors and with many different subtypes. Serotonin receptors, 5-HT is serotonin. Again, metabotropic and range of functions and distributions in the brain doing different jobs. Finally, you get your histamine receptors, again, metabotropic G protein coupled. The histamine receptors 1 and 2 are postsynaptic, whereas histamine receptor 3 is exclusively found in the presynaptic membrane and has this role as kind of an autoreceptor, shutting off histamine release and therefore kind of providing negative feedback. Now, whilst I've explicitly pointed this out for histamine because it has this interesting quirk, actually you will find that negative feedback is quite a common feature for many of these neurotransmitters and, you know, is important at many different synapses. Uptake is via their respective transporters here, dopamine transporter, noradrenaline transporter, serotonin transporter, and there's also a role for monoamine oxidase enzymes to recycle the monoamine group and enable the synthesis of more neurotransmitter. Loads and loads of drugs acting on monoaminergic transmission. Most of these you will come across elsewhere. Things to think about is that your psychostimulants are all acting through dopamine. Your psychedelic drugs are through serotonin, but note as well that there's kind of this broad function for serotonin in terms of this 5-HT2 receptor, which has lots of widespread effects, but then there's 5-HT3, a different receptor that has a really specific role in kind of regulating vomiting, and therefore it can be targeted by antiemetics, which are drugs that stop you from throwing up. L-DOPA, I'm sure you've met that before. SSRIs are your most common antidepressants and anxiolytics now, which work by increasing serotonin in the synapse. Remember that the earlier class of antidepressants and anxiolytics were monoamine oxidase inhibitors, and you might come across those at some point. Continuing on, what are monoamines doing in the CNS? So what is characteristic of these cells and these types of neurotransmitters is cell bodies that sit in the subcortex, and then these really long diffuse projections, really long axons projecting throughout the cortex and interacting with many, many neurons on their way. They do this via these kind of swellings in the axon that we call en passant junctions or varicosities. And what sort of things are they doing? So noradrenaline and serotonin are really important in regulating your alertness and your awakeness. So they sit down in these nuclei, the raphe and the locus coeruleus, in part of what we call the reticular formations of the brainstem, and they kind of send signals up to regulate our alertness and wakefulness. And dopamine, again, will be covered extensively elsewhere, but dopamine is really important for processing of reward, or should I say reward prediction and reward prediction error. And that is via the ventral tegmental area projecting into the prefrontal cortex, as you can see here. Also, remember that really important role for dopamine in regulating movement through the basal ganglia, and those nuclei, you know, the substantia nigra pars compacta, which is part of the basal ganglia, sits pretty close to the VTA here. So they're the two roles for dopamine. Couple more to do. Cholinergic transmission, we have met this a lot during preclinical, during year one, and it's exactly the same sort of mechanisms as you see in peripheral nerves. So choline and acetyl-CoA, synthesized by choline acetyltransferase. You've got an acetylcholine carrier for into the vesicles, and then you have your nicotinic receptors, which are your ion channels, and your muscarinic receptors, which are metabotropic, and they are a big family with lots of different G proteins and lots of different roles. You have acetylcholine esterase in the synapse, breaking it down, and a presynaptic choline transporter for recycling. And similar to the monoamines we've just met, cholinergic nuclei are part of this reticular formation, as well as noradrenaline and serotonin, and these send these diffuse connections up into the cortex, which help regulate wakefulness and in particular arousal from sleep. Last slide, I promise, and I want you to, this is just something for you to kind of think about, maybe particularly with respect to other aspects of your neuroscience course, but you learn a lot about synapses, and I need you to think that synapses are very complex, and it is not just a case of kind of like one input equals one output sort of thing. And there are actually many different computations and lots of processing that happens at the level of the synapse. Some examples of that is this idea of synaptic integration, which is where you get numerous presynaptic inputs of the same neurotransmitter, of different neurotransmitters, onto a single postsynaptic cell. So there's going to be a computation process going on there. You can get kind of the summation, the adding up of these inputs over a given area, like a given part of the dendritic tree, or a given time period. You get neuromodulation, which is the idea that you can have numerous different neurotransmitters and neuropeptides acting on a single synapse in order to kind of fine tune and nuance the function of that synapse. And that can be via the release of co-transmitters, which is where you have two neurotransmitters being released at a single synapse, or by this idea of bulk transmission, which is where actually when you're releasing lots and lots of neurotransmitter, it diffuses within the CSF and can actually have more widespread effects than just the single synapse. And finally, you get synaptic plasticity, which is kind of changes to the strength or the gain or the volume of a single synapse, and that takes place via LTP and LTD. And that is it for this lecture. I am sorry for overrunning. I was 7 minutes over, not 5 minutes, which does make me sad. I am sorry. Please follow this link or the link that I have shared into the chat to complete the feedback form. This is the first lecture I've ever given. It might show, I'm sorry if it does, but I would really love your feedback, positive, negative, constructive, hopefully, for me to improve. And also, if you complete that form, there will be some extra questions to go over the neuronal function slides that we've just looked at, and also an opportunity to receive this handout, which I hope will be useful for your revision.